Thank you, Rainer, for inviting me to come here and give a talk, and then thank you very much for a nice introduction. It's a great pleasure for me to come here and to talk about our research and give a colloquium. We do biological physics within the physics department. Right, we have people doing experimental and also theoretical biological physics. Biological physics is very broad, ranging from molecules, cells, to brains, to systems. So our lab is focusing on single molecule manipulation, and we also did some nanoparticle aggregation. We're using these tools to probe biomolecular interactions. So we'll talk about that. So here, I'll first outline my talk here. I will start with the background and the motivation. Why do we do this type of research, and what we try to, we expect to see, and what kind of things we have seen from these systems. And the experimental technique is mainly we're using atomic force microscope. Most people are familiar with that. And that's used to manipulate a single biomolecule. And we're using this, and we use a recently derived theory, and it's a non-equilibrium work theorem, to convert these non-equilibrium measurements to equilibrium information. And these are important for biological systems. In these force, we measure the force. These can also be used as a probe for probing the dynamic states of biomolecules. So molecules are changing. They have the equilibrium states, but they can also adopt different states, and one can have some information with some of these techniques. And I'll give you two examples. The first is a discovery of a novel state of a PolydA. It's a single-stranded DNA composed of just a base. It's one type of a basis. And the second one is a shear-activated molecular glue. It's a large molecule present in human bloodstream. Then I'll conclude the talk with past and the future. So I see a lot of students, so I would also talk about a little bit how we started looking at these, what we're trying to see in the beginning. So first we look at single molecule manipulation. That was this technique was invented in the 90s, and people started to be able to use these nano techniques, nano tool to graph a single molecule, usually relatively large biomolecules. They grab them and pull them. So the point is one can observe the entire trajectory. So unlike other type of a biochemical, biological techniques, usually they probe state-to-state transitions. So if we look at like quantized state, the biology in biological systems, the biomolecules also have a different conformations, like structure, different structures, means that they have a different energy state. And then if one can trace that and looking, it's changing between these two states, so one can see how the molecule is doing. But in the single molecule manipulation, we're actually seeing the entire trajectory. So it's not just jumping from one to another. We pull the molecule. Depending on the molecule, we could be unfolding a protein, or unstacking, or melting a DNA, or ripping apart complexes. But we're seeing the entire trajectory from the beginning to end. And then, of course, it comes with a problem. How do we interpret that? So originally, the motivation as a physicist is to understand this process. So one can see many things coming out of these biological systems. But how do you interpret that? Do they actually know what they're saying? Do they actually say something is representative of what they've seen? And so in using a theory, one can reconstruct the entire free energy landscape from these measurements. which is very fundamental in biological systems, and I'll show you how one do that. And conformational changes during these transitions can be probed if one could focus on certain states. We'll show you an example. So the basic setup is relatively simple. AFM has a tip. It's basically like a fishing rod, and there's a substrate that's a surface. So for AFM, most of the time we rely on non-specific binding, which means we just spray these molecules on the surface. So they're sticky enough, right? And you know, if you have proteins, things, if you don't use the soap to rub it, it will stick to the container, right? So to some extent, part of the molecule will stick to the surface. Of course, one can modify the surface and add some functional group and make it more sticky. But anyway, that's the basic. And then part of the molecule will stick to the tip. So the tip really comes down to the surface. It's literally like fishing. You go down and pick up to see if you get anything, right? It's like fishing. If you don't get a fish right away, you may put your fishing rod, let it sit there longer, right? And then when you feel something, right, so there's some resistance, you pull it, you probably have something. So we go, the tip goes down and pull up. If there's nothing, then we see no forces on it, right? So the tip is going to tell us that there's nothing, you didn't get any fish, right? That enough. But if there is something resisting, and you can feel it, and the force extension curve is going to tell you, now you got something. So with that, so the analogy is somewhat a basic idea is, if we look at the molecule, right, we don't really see the structure. The structure usually is solved already by other techniques. So usually we start with molecule, we know what that is. But now we want to see the interaction and then how strong it is and what it does. So we pull it apart. So it will tell us something, and it depends on the situation. We need to know how to interpret that. So for example, if it's hard to pull, then you know the molecules are tightly bound together. So right, so it's like a fish. Even you don't see it. If it's really hard for you to pull up, it's probably a big fish, right? And sometimes it's not a big fish. Maybe it's some fish who likes to fight, right? It's also possible. So there's several interpretations, but overall, it's telling you some high resistance. So you keep pulling, and then you record the distance curve. It will tell you something about the molecule, right? It doesn't tell you everything. It does not tell you the shape about the molecule. But if you know something about the molecule, and then you can guess from there. For example, if you know a shark always does a certain bite, and then now you pull it, it bites like a shark, then you can somewhat conclude that this molecule, well, it's probably a shark, right? So but you need some other information. You don't really know it just by pulling. You know it from you know before. It does like that, right? But maybe it's a shark, but maybe it's a baby shark, right? Or maybe it's a sick shark. It's a little bit different. So when you pull it, sometimes there's some variation. And we need to know when do we say that's like that with some variations. So there's certain conditions, and then what's happening is some interesting things happening with the molecule, or this is something, or actually we get some junk, right? So maybe you get stuck and you get some seaweed there stuck there. Yes. Yes. Okay, so if we don't touch the surface, then it's just zero all the time, right? So the cantilever is not bent, right? It's reproducible. Once I yes, a good question. Yes, that's one way to do experiment. That's yes. So that's actually a very good way of doing it. If one want to make sure you get the same molecule and going back and forth. So that's commonly used. Yes, we get reproducible results. But one thing, sometimes we want to do molecule, like we pull it, like maybe we unfold it, right? So we let it make it relax. We rely on the protein itself to refold. It may or may not refold properly, right? So like 99% of the time it will refold properly. So we can do it back and forth. But sometimes if we want to start with something definitely folded properly, then we may want to pick up another one. And these will have very similar curves. So we have pure, just one kind of molecule, and then there may be some kind of junk on the surface. But the non-specific signal, like so-called junk, is not reproducible. So if we keep doing it, we will see something is reproducible. We analyze those. So the things that are not reproducible, those are thrown out. Right. But if we have one molecule going back and forth, then that's almost always reproducible, unless we don't wait long enough, it didn't have enough time to refold to its original state. Right. Or the molecule simply get too tired. So another method, which is also very popular, that is involved optical. Optical, this is actually not in, well, it's trapping, I would say. It's laser optical tweezers. And so we see the laser beam hold this one beam, and the other one could be held either by another laser beam or by a micropipette, and then pull the molecule. So these are functionalized molecules get attached to the two ends like that, and then we pull, and the molecule will experience some force, and it'll drag us, move this beam away from the center, and this harmonic potential will tell us how much force is on the molecule. So we can also then force distance curve. So that's another way of getting it. So these are the basics, and then with the experiment and the theory is the non-equilibrium work theory. And to make it a long story short, here the molecule, we're usually interested in free energy, right? So each conformation of the molecule has a certain energy, and its free energy tells us the stability and the interaction. And so when we pull it, this is a mechanical force. So in principle, if we think about that, if we know the force we put in, and we know the distance we pull, we know the work we put into the molecule, right? So that work is related to energy, right? How? That's a question. So for the second law of the thermodynamics is the work we put in is larger or equal to free energy. So we will waste some energy if we pull it fast, right? Friction, heat, all these dissipation. If we pull it, unless we pull it infinitely slowly, right? So the molecules are almost always at equilibrium state. Then the work we put in is equivalent to the free energy. But then that means literally the experiment should take forever. And in 1997, Chris Jasinski derived now so-called Jasinski's equality. It says you don't have to do the experiment infinite slowly. You just have to do it for infinite time, of course, and there's no free lunch. But anyway, in reality, and if it's large, it does not necessarily need to be infinite. But anyway, using the work, I put it in this exponential averaging, then we can get a real free energy. So the idea is for these small systems, even though, so the most of the work you put in is larger than the free energy difference. So now you think, okay, if it's always larger, no matter how you average it, right? As long as you don't subtract anything, it's always going to be larger. But for small systems, of course, with all complicated derivations, I'm not a theorist, but for small systems, the fluctuations of the thermal fluctuations, basically, so the single molecule, for example, in the solution, there is a thermal energy from the solution that sometimes it could be borrowed by the molecule. So the molecule sometimes requires less work than is required to do certain work. So sometimes the work we put in is actually less than what is needed. Of course, those situations rare events, but when we put it into this exponential average, the rare event, when this W is smaller, it's weighted much more heavily, right? So overall, it will give us the right answer. So from the theory, it says this equation is exact. As long as you start with the equilibrium state, Yes. Oops, sorry. When I do that, oh. Okay, so there was a way that if the long answer is a short answer, but I don't think you want to know the absolute detail of the equation. So I can show you this. I was not going to show you this. The histogram method. That's exactly what we put in. But in principle, well, I wasn't trying to show it was like the slide. It was the high slide there. But I can. Why don't I use this one? Okay. Okay. I'll go through this. So let me just. Okay. Let me start with the system. I'll tell you how we do that. At least conceptually. So this was the first test by Bustamante group with RNA, the genesis equality. So it works. So we're just trying to apply. We don't really want to test it. In our opinion, it's the error bars for single molecule usually is relatively large. We know it works, so we can apply. I think testing it's a little bit pushing it. But however, this has been tested in many different ways, not just in bio. So it's pretty much well accepted. It works and it's correct. So we want to see how one can get the free energy landscape of protein folding from these. So the first thing we try is Titin. Titin is muscle protein. It's a well studied. People know the structure, some of the structures and what it's like. So this is a muscle, and going from Z line to M line, the striated muscles, and the Titin is this thing that holds the structure together. So it's not Titin is not the one that's doing the contraction and relaxation. It's a molecular motor, as many of you know. Molecular motor is the one that does the muscle contractions and walk on these filaments. And Titin is just holding the structure together. And there are many, many domains. We're studying one of these so-called I-27 domains. Okay, so the I-27 domains like this. So we have these engineer domains. So these engineer is domain. Each domain is from Titin, so muscle protein, and then engineer to make 7 of them link. Actually, 8 of them linked together. They're identical. So we pull them. So these molecules behave more like a rubber band. So it's like that. And the cantilever behaves like spring, hook spring. So it's represented by the spring here. So we pull it, and then we measure the force extension curve. A typical force extension curve looks like this. So if we analyze one of these, so each one we pull it, and then stretch it, and then it breaks the next domain in the force drop. And then we can fit these with polymer physics model, warm light chain model. They really look like a flexible rod and follows this equation. And so we can look at see what happened in this process. So here is the answer to the question. So this is the force, and this is the extension. The very approximate way is the force times the distance is the work that's put in to pull this molecule from here to here, right? So if we integrate the whole thing, it's basically like the area here is the energy, free energy requires to go from this state to that state. In reality, Jasinski's equality actually it's a little bit more details. I'll sort of skip through it unless you really interested in all the details. But anyway, so it's more or less like FDX, but not exactly because it's still, because when Jasinski's equality to be applied to this, it's average in the time space. So this is the distance. So they're not completely one-to-one relationship, not exactly, but approximately. So, but anyway, it's you will use the binding method. So it's similar to we have many of these, and then we average the FDX. We just get all the FDX and then average it. In reality, it's done in a time domain and then converted back to the X domain. But what happened is we got the curve look like this. And so this is this process. I'm going to show you. This is an animation. It's not a molecular simulation. This is basically a movie, the cartoon from my student made. It's like this. So it's a Titin, I-27 domain. We lift it off the surface, and then we pull it. So now this is the highest force, and then these bonds are going to break. So at the peak, once it broke, that's the peak. It's breaking. That's the peak of the force, and the force will drop. But then we keep pulling it to pull it all the way straight. Okay, so this is so that the second part of the movie is actually showing this. We pull it straight, and then until it's completely straight, and then the next domain. Now this the whole molecule is under very high force, high tension, and the next domain is going to break, right? So basically, the weakest link is going to break, which is going to be the hydrogen bond domain, because the covalent bonds are much stronger, much more strong, stronger there. So once the hydrogen bonds broke, that's considered unfolding of the protein, and the force drop. And then now we're probing the next domain unfolding. So this whole part is called the free energy landscape. So we can get that through this, and we can integrate it. So one will view that. So to compare, this is a little bit of a detail just to show we can compare to data measured with other techniques. So other techniques usually measure the kinetics, K here, the rate, and then this number K0 pre-exponential factor is usually guessed. And so this is the estimate, so they have a delta G is estimate. Versus we are measuring delta G directly, although the error bar, especially largely, is coming from the X. So anyway, however, we got a number on the order of 11 kcals per mole, and then people use the kinetic measurement. They have estimated numbers between 10 and 22 kcals per mole. So it's consistent. However, it tells you nobody knows exactly what the answer is. So it's still worth getting new techniques to measure these quantities exactly. So first, for us, we're trying to do, in the beginning, we're trying to get equilibrium information from non-equilibrium measurement, because we want to make a good use of the non-equilibrium work theory, right? But once we apply to the biological systems, in this particular case, it's about protein folding, right? So the general public, protein folding. So why is protein folding important? Because protein folding, if you know how protein fold properly, you can know why it folds, misfold, right? So misfolding or protein misfolding will some diseases are caused or related to protein misfolding, like Alzheimer's disease, some neurodegenerative diseases like Parkinson's and mad cow. So one would imagine if you know exactly how protein folds. So protein, when it was first synthesized, it's a chain of amino acid, like the stretch to the molecule I just showed. It's a long chain. And it doesn't have a proper function unless it's folded to its correct three-dimensional structure. So how does it do that, right? So the protein has so-called folding energy landscape. So it's like the landscape. You go downhill, the protein is walking on these energy landscape. They want to go downhill to the bottom of the hill. And then so if you know the landscape, then you can predict a few paths for the protein to fall, right? So you can also see maybe what kind of situation or how the protein could deviate from its correct path and to misfold, go to the wrong path and get stuck in a different state, the wrong state. So, and of course, in the disease, it's always a lot of things involved. It's not just one thing, not just protein misfolding. It has to require that they get into the beta sheet and then deposit, form the fiber and deposit in the brain, like that. But anyway, that is the fundamental things of what happened in the molecular level that's related to disease, is protein misfolding. And then, of course, we have even more general topic everybody wants to know. Protein, how does protein fold? How do they misfold? Can we use the physics technique to do that? Can the fishing rod tell us about all types of disease, right? So it's all very exciting. So while we're doing very sophisticated physics, it's also possible to make it sound very general and interesting to the public, so we can get support to do more physics, right? So it's also fun. So this is the landscape I was talking about. So the protein, what happened is people study it many different ways. So what we did is we start with a state and then go over the barrier, we pull it over and make it unfold, and then we keep stretching it, right? So the different techniques are probably different things. For like fluorescence, usually look at the folding, protein folded to unfolded state. They look at these states, and then there was a question of if these techniques are measuring the same thing. I'll skip the details, but the conclusion is pretty much they are probably not exactly the same. The different techniques are probing different pathways, but they're probably similar, at least initially. But the manipulation polling technique will probe all the way to this state, which is never seen with other techniques. So by dragging the molecule, stretching it, one can actually see the areas here usually not seen with other techniques, which I'm going to show. It's actually telling us something in the DNA. So there are different applications we can think of, and we can look at fundamental biological problems like a DNA mechanics. And it's just a very simple DNA, but it's like a polymer-like molecule. And then it's more like into medicine, we study for Willebrand Factor. It's a very complicated, large molecule, and we'll go into a little bit of the story about that. And then other diseases like protein is another large molecule that's involved in cancer. And the other biomaterials, like one can even stretch graphene and look at it. I'll show you mainly, you'll talk a little bit more about the first two examples. So overstretching of PolydA, okay? So we have DNA. DNA is a double helix, right? We just earlier have the conversation. One of the big discovery done by physicists, which was Boston Creek, in the field of bio, is the DNA double helix structure, right? And the DNA diffraction pattern, the X-ray diffraction, is arguably one of the most beautiful experiments done in the last century, right? So whatever you did in this century is a new thing, so you don't have to worry about being compared to that. But anyway, it's really a very important discovery, the DNA double helix. It's very cleverly designed. So the DNA has bases that carries a genetic code. Okay, so the sequence of these bases tell us what protein it makes, and they usually, well, they are stored in the double helix form. So when the process needs to be something needs to happen, like for example, it's called transcription, but basically one needs to get the code from the DNA and need to read the code. Now we want to interpret the code to make proteins. So first we need to open up, right, to read the bases. And so the DNA needs to be the structure of the DNA needs to deviate from its original form, right? So it's the mechanical property of the DNA is important in this process. So how is it got read? And also there are other systems like a virus, influenza A virus. DNA is, or RNA could be coated with proteins and they self-assemble into the virus. So they have to be stable in the form when they're interacting with proteins. And they usually are packed, very highly, densely packed, and to form, and they will actually self-assemble. So it's not necessary someone stick them into a ball. They actually self-assemble, have things wrapped around it. So it's basically the free energy is controlling them to do that. So it's no external force to push them to do that. They actually like to do that form. So it's free energy is always important to in terms of determining these stability and how these things take place. And then also base stacking is important. This is so-called base stacking. When they form these double helix structure, the bases are well aligned and they form a regular structure. And that energy is important for many functions. And also in PolydA specifically, A base has a lot of interesting properties. So let's just go back to the basics of the DNA. DNA is composed of four bases, A, T, C, G, these four nucleotides. So the combination of these, the sequence of that is the genetic code. And the double helix is the one strand of DNA and another one here. And in between them, they form hydrogen bond. So C always pairs with G, T always pairs with A. So that's very interesting and very important in terms of a double helix. So you have one strand. Because of a base pairing, you know exactly what the other strand is, right? If you have a G, the other one has to be a C. If you have an A, the other one has to be T. So you only need one, you will know the other one. So that allows for replication, duplicating the DNA. So we have lots of DNA repeats, copies of DNA. And then also allows these transcription. So when anything needs to open up, you just need to read one. And you're also able to make another one based on this. And then you make another one and you open up and you get more of them. And also these bases are hidden basically in the middle. So it's, so DNA is very stable. So in the beginning, so people trying to solve the DNA structure was pretty hard because people knew that DNA carries a genetic code. And you need to read the code. So naturally, they think the basis has to be out, so I can read it. So they try to come up with a structure with the basis outside. They just couldn't. So and just you look at the X-ray structure and all that. It just does not make. It just doesn't fit. Nothing fits the experimental data. And eventually, they look at the chemical. They decide, well, just start with the chemical structure and then see how you form bonds. And then they realize that these are just matches nicely. It's like the lock and key. That so the bases is actually inside protected, and only when they're needed, they're open up, and then you get rid. So it's really a very clever design by nature. So PolydA has some unique properties, and here I will first talk about the in 2007 Maslach group at Duke. They discover when we look at the DNA. Now I'm skipping a little bit. So DNA, if we poll it, it has these double helix. Okay, so if we pull it, even without unzipping it and just opening it up, we just pull it straight. You can imagine if you want to form a perfect double helix, it has to be exactly that structure with that angle. If you pull it straight, you have to break all the hydrogen bonds in the middle because they can't really keep that kind of relative positions anymore. So in reality, you open up the bond between the two strands, actually they're melted. So with the single strand, you also disrupt the base stacking that way. But usually, single-stranded DNA does not start with the base stacking. So you stretch them, they're all like a rubber band. So it's like the blue curve, like this. But PolydA is unique. If you stretch them, first there's a plateau here. It's obvious, but then there's another one here. So what are these? The first guess was the PolydA actually has base stacking. So in the beginning, even it just by itself, it does have these helical structure, even though without another matching bases. It's like the staircase just going regularly going up because it has a relatively strong base and base interaction. So when we one pulled it, at first it would disrupt that stacking. What happened to the second one? What else could it be? So they found this, and then so we're trying to see exactly what's happening. So there are several proposals. First, we say, oh, maybe there's sugar is having a different structure. So DNA is quite complicated. In addition to these bases in the middle, there's a backbone that holds them together. And outside the backbone, there's the sugar, right? So the sugar could have a different structures. Whether it would change these force curves is not known. But imagine that, but then we would like to see if there's any evidence for that. So what we did is we changed by changing some of the conditions, the polling speed and cell concentration, some more physical parameters. We actually saw two pathways. So we going here, we could go this way or this way. So the blue curve is a higher energy because it's a higher force, right? So force times the distance. So pretty much this area tells us how much energy we need to put in. And then relax, when we let the molecule relax, it usually comes back with this lower energy state. So one can actually go back and forth. So that's the question. You go back and forth. So we actually pull on one molecule and go back and forth. So it could actually go up and back. So depending on how fast we pull it, sometimes there's a hysteresis. So we go up, we could go to the blue line or the red line, depending on how fast we pull it. And coming back, if we go up with the blue line, it would turn around real fast. Sometimes it gets stuck in the blue line and come back. But if we wait, usually they come back with through the red line. So it's a lower energy state. And then if we match that with other double-stranded DNA, we see the matches pretty well. Here, there's something happened with the double-stranded DNA melting, and also there's a little bit more of details here. So the PolydA could even go smoothly like this. It's almost like a rubber band, or it has these two steps. So what do these steps mean? It has to mean that they have a different structure. So the conclusion is it must start with the stack structure. And once they go over this, the stacking is disrupted, but it goes to another stack structure. So a little bit more loosely stacked, but still stacked. And once it reaches this 0.6 angstrom nanometer per base, it goes to another stack structure. So there are actually 3 different ways of stacking depending on how far apart the bases are stretched. So this area was never probed. So people know PolydA you could unstack to a different thing, but it was never obvious whether it will go to another structure, another stack structure, or it goes directly to a random coil, or let alone there's another stack structure. So this was only visible with these force curves because it was not really, these don't really have very clear signal, a chemical signal for people to detect. And also in other chemical methods, people don't usually stretch the DNA that far, so they never seen this. So this is one interesting property this technique allowed us to see. So that's a summary of this part. So the PolydA free energy landscape must be look like that. So there are 2 pathways. This is a random coil. This is stack state. In between there is a a barrier. One can, if one star here get trapped here, it will go over this way. If it allows enough time to go through equilibrium, it will go to the lower energy state. Okay, so with these multiple, okay, so just to satisfy a few people's interest about DNA particles, I'll mention a little bit related studies about DNA melting. So this is a little bit more biosensors. The colloquial gold particles, people use them, they're covered with DNA. And the DNA, this could be used to detect different things. That for a while, during that time, the insects was a little bit of a hot topic, so they used that to detect insects. Basically, just to look for certain DNA sequence. The basics is like this, if one can see. So it's like a DNA microarrays, it's called gene chips. So each of the spots here represent a particular DNA sequence. Okay. So these are known sequence deposited on the surface. Sorry. So that's the basic of the gene chips. So the deposit, the DNA sequence on the, this is known sequence, and then we wash some DNA target. These are sample, like something we want to analyze and see if there's insect DNA there. They bind to that, right? So the ones that comes has fluorescence tag on them. Then we shine the laser. The ones, the sample, the DNA has certain sample, and then it will shine there. Then we know what kind of DNA it has. Okay, so that, and then so this we rely on DNA hybridization, which is the DNA base pairing. So we look at the colloidal particles with the DNA attached to them. So the blue and red, if the DNA, the sequence are not complementary, they will not form double helix, which means it's called hybridization. So if we have a target DNA that's complementary to this part, the red, the other half is complementary to blue, and it will bind to both of them, right? So the sequence match, they will bind to them, and then we'll link them together. If many of them, then the particle will aggregate. And yes. Okay, this is just man-made. So it depends on the sequence you want to test, and you design what sequence you want to detect, and then people deposit on this. So it's either synthetic. Sometimes they use a mask to synthesize the DNA with like 30 bases, or sometimes they use the long one. They just use UV to stick that to that. So just the idea is just to know there's some known sequence. I want to see if this sample has that sequence. Then I'll just deposit that specific sequence in a specific spot. So if the spot later on is lit, then I know this organism has that sequence. It's just a design. So you design, of course, there's some kind of redundancy in these sequences, but you design a sequence that's going to test a specific part of the DNA in the seed. Right. Yes, yes. Right. Right. Okay. So for each one, I don't know if that's your question. So each one, in principle, it has to be completely matching for that to bind. But of course, everything has noise, right? So that's why we want to study the quantitative thing I'm talking about. So that's bringing up a good point. Because sometimes to say if there are 30 bases, what happens if 29 of the bases match? There's only one mismatch. Will it bind? So usually it will, but with a slightly different melting temperature. So it may change the stability. And of course, if we study more, then we see, depending on the mismatch and deletion, all different so-called defects, sometimes actually increase the binding, so-called binding constant, if you can define it that way. But in principle, the idea is if it's the same sequence, it will bind. If it's not, it's not. And then the noise level, then we'll have to worry about quantitative later. So that's what we want to be quantitative. So for example, we're just setting some examples showing, okay, exactly like I said, it's not so obvious if it's the same bind, if it's not the same, it doesn't bind. And it's not even so obvious if you have a mismatch, which we call a defect in physics, that's necessarily going to lower the melting temperature. So, so this is what it aggregates, and then we will see in the solution, this is a red solution, and then when it aggregates, it will turn purple, and if it aggregates a really large aggregate, it will precipitate, and the solution will become clear. And so that's from this technique, instead of using laser, we can actually see from our naked eye the things are being aggregated. So when it aggregates, that means that it has the DNA that's complementary to these. But if we look at the more careful, some unique situation, for example, if we look at all these eggs, so in the solution, if we have a longer DNA with more bases, with the more base pairs, the melting point is actually larger, higher. So if we look at this is the probe, okay, it's all A's, and then the linker, so-called linker or target DNA, source C. So this one is the black one's the free DNA that comes in and binds to them. If they're shorter, so the total is 24 bases. This is shorter, like 23, 22. So in the free solution, the shorter it gets, the lower the melting point will be. But when it's bound, these DNA attached to the gold surface particle, and then it's also sometimes similar with other base surfaces. The melting points actually will change unexpectedly. In this case, it goes like a zigzag, up and down. So what we found is if the target, the linker bases, if it's has on a different number of these, so one is 12 bases, one is 11, that means these two are a little bit different. They'll have a different lower than expected melting temperature. So it just says, in addition to, sometimes the free solution DNA thermodynamics, it's not necessarily so directly you can just apply that to the surface DNA reactions. So that's actually another reason the gene chips has a lot of noise. People actually are aware of that. And like you said, sometimes there is a mismatch. And depending on where the mismatch is or deletion, like if there was a missing pair near the surface, sometimes they actually increase the melting temperature, make it bind even more strongly. So that needs to be taken into account. Okay, the last one is an example of the more medical von Willebrand factor. Von Willebrand factor is a very important molecule in the blood. So it's a giant ligand, so one of the largest molecules in the body. It's important in initiation of the blood clotting. So that's the first molecule. It's pretty interesting. So we have the arteries and the vein. The stop bleeding, the mechanism for stop bleeding is a little bit different. So the artery, if we have a cut and then the blood, it shoots out, right? So you need to have a very fast response and then things to stop the bleeding. So the von Willebrand factor is a shear-activated molecule. So what it does is, once it's activated, the platelet will bind to the von Willebrand factor. And the activated platelet will activate other platelets, and then they'll form a clot and then stop the bleeding. But the von Willebrand needs to be activated because it's circulating in the blood all the time. Most of the time, normally it's not activated. Otherwise, we'll have all the blood clotting everywhere, and we will not be alive. So it only activated when there was, say, like a turbulence or like a shear. So like usually the blood flows like this, and then now you have a cut. Right? And then now the flow is turning. Then that creates an extra stress, and then the molecule gets activated, binds the platelet receptor. So this is a von Willebrand factor here, and then this is a platelet, this is a cartoon. So the von Willebrand factor binds to this the receptor GP1B alpha on the von Willebrand factor specific domain is being activated. And then von Willebrand factor is pretty unique. It's usually very long. It's synthesized and very long, and it gets cut when it comes out that synthesis cell. And if it's actually long, it's actually hyperactive. So it actually needs to be cut to be inactive. And when it's normally it's inactive, and it gets activated by shear, right? So it does not want to be always activated. That means it's hyperactive, or it doesn't want to be cannot be activated. And there's some story about this molecule. It's a little bit difficult to study because its activity depending on the size. So if you chop them off into small domains, and most technique does, they don't really have the same effect. So we have to study the molecule in intact form. This giant molecule has 12 domains per monomer, and this molecule has many modes. It's a multimer. So really, it's a huge domain, huge molecule. And the A1 domain is the one that will bind to the platelet, stretch the molecule, and then see the unfolding pattern. So what we found is we have many multimers, but most likely there's only one domain that can unfold because the rest is disulfide bond, and we usually cannot break the disulfide bond. And we see these unfolding force somehow is correlated with the activity. So the inactive form unfolds at lower forces, the activated form, the shear form, and the ultra-large form get unfolded in the higher forces. And in knowledge, we just use the force as a marker to see which state they are, because it's almost impossible to tell with the other method. They don't really have a fluorescent signal depending on the state. So, but the force is different. Then we can track the force and see actually the force will change. So the molecule is shear activated. And once it's activated, it unfolds the higher forces, and then it will relax back to its lower force. Okay, so during the course of several hours, a little bit, 10 hours, eventually reacts to that. So this is pretty interesting because people used to think some of these processes may only last very short time. A few hours really is much longer than what people thought. So what happened here? From these, one can see the inactive form is a plasma form of von Willebrand factor is inactive. That's the one that's circulating in the blood, the regular form. And this is the activated form. Once it's put into a high shear. Actually, we put in, this is a detail I sort of skipped. We put in a very, very high shear, much higher shear than usually even the wound experience. It's a much higher shear. This is almost like a pathological level of high shear. So we unfold it. It must happen this way. When it unfolds, it goes through a different pathway. So versus the inactive form. So these two forms are different. So it goes through these way. But if we pull it very, very slowly, this molecule actually we're able to get back to the original form. And then we can unfold. So when we pull it very slowly, we can see the unfolding force is almost the same. So this is coming from the force studies in which it tells us we are using the force. Here we're not using the force to introduce the shear. Here we're actually using the force to probe which state they are. So it's a unique way of looking into that. Okay, so some small things in a few examples for hurricanes. It's long. We'll just go through it. So very fast. It's a long multi-domain molecules. It's involved in many of the processes. And then we can stretch in and look at these bending, basically persistence length and how stiff it is. And then other things in biomaterials, and I'll not go into detail. So the graphene nanoribbons are also long nanomaterials with stretching, and we study the mechanical properties. So here I'll summarize what we talked today. I've gone through the single molecule, basically single molecule manipulation. What it does is that it allows us to observe the entire trajectory from beginning to end. And then the stretching, sometimes it goes through different pathways, and it will be very obvious from these force curves. And the force can also be used to track different states, and the molecules will tell us something about the mechanics of these systems. And last one, how do we know if this is any good for anything? So we'll just use an example. Before I was doing single carbon nanotubes a long time ago. It was discovered in 1993, the same time with Hiroshima. And at that time, now it seems obvious, okay, nanotubes and nano is everything. But at that time, people asked me, do you think a nano is going to sustain? So you never know at that time. It could just go away, but it could turn into something very big, very important. And now you see nanotubes on the cover of the chemical catalogs and the books, so it's everywhere. We're selling it. So it didn't go away. So even the discovery, when you first see something, it may not seem useful and may not seem to have anything to do with anything. At that time, it was not even obvious what kind of name we should give them with the nanotube with single layer, atomic layer walls, or some kind of description. But it may be, so that's what we do physics. We try to see if there's anything, try to discover something. It may or may not, and then I'm pretty sure many of the things when discovered eventually didn't turn out to be anything. But that's okay. It satisfies our curiosity. Something will turn into something, okay? So I want to encourage all the students and postdocs here. So if you see something, you always keep hope. Something may turn out to be very important. And then the important thing is you go look for the next one, right? And if this one it's not going to be the earth-shattering, or maybe the next one is, okay? Just keep trying, never give up, okay? So we'll go with, finally, thank the students, postdocs who actually did the work, and the funding agent, my collaborators. And lastly, thank you for your attention.
For the misfolded proteins that are thought to be a key issue in neurological disease, is the free energy of the misfolded state higher or lower than the free energy of the healthy state?
So by definition, it's almost always higher. That's the native state is pretty much always the lowest, right? So what happened is that's a little bit unique situation. It's like it's a beta sheet, so it instead of folding to like a globular, it's like a long fiber, and now you have bad influence. So one misfolded protein makes other protein misfolded, right? So we don't want to have a bad influence. One person doesn't study, the other one doesn't do it either. So it makes other protein misfolded, and then they all become this long, and then they condense. Once they form fiber, the fiber form is actually pretty stable relative to many of these. Right, but if you compare one, it should not, it started like that, but once it formed like that, and then it's very stable.
Right. To follow up on that question, suppose I have a cantilever standing in front of a microscope, and I'm trying to see the force. Should I think of this as a derivative of the free energy with respect to position? That's the force, correct? Not the derivative of an electrostatic potential, right?
The way we do it, let me see. We always probe the free energy, so it's a derivative of free energy. Because we probe mechanical force. I don't know if I can think of a situation if you can possibly probe the electrostatic forces. It's probably unlikely. I think most of the time it's free energy and then using the forces, so derivative of free energy.
The question was maybe more about, maybe it goes back to this theorem that you showed, the connection between work and free energy.
Right.
Because I think you came in after you showed that. You wanna, so the connection between work and free energy, was that what the question?
What was the question? I'm not sure I understand the question. So
I think he was asking about the connection between work and free energy, which you had the theorem,
right, Jablonski's, right. Yeah. So are you worrying about we ignore the electrical energy, or
how can you get the free energy out of it?
How could I get the free energy of the work? It's basically FDX, but it's pretty much just because here we're looking at the free energy, and then we're looking at how much work we put in. We're only putting in mechanical work, so we only use FDX. So that's all the work we put in. If you use other work, then you can, and that will turn into the free energy. Then you may calculate whatever work you put in. But I'm in this type of setup. I did not introduce any electrical current, so I don't have other type of work performed on the system. So I only do F. Works. Okay.
Throughout your talk, you were stretching things, sarcomeres, DNA, nano sheets, fullerene sheets. I wonder and you showed many graphs, what is the force constant in all these graphs? I couldn't see the force constant of the spring models.
Oh, okay. So, well, so here we're fitting this. Well, they're all pretty similar, right? So they're all fitting this with the Walmart chain model. So, okay. Oh, piconewton versus nanometers. Right. So we just fit that with here we assume they're so-called non-extensible, so we can't really stretch them. They're infinitely stiff in terms of stretching. So we only consider binding, then we fit it with this model. Okay, so we have X, have the force, and then we get the persistence length and the contour length. So we just basically get the bending module. We can add another term to take into account once they bend, we stretch them up to their contour length. We keep stretching them because they're stretchable. That's another additional term. But usually they're relatively stiff.
So they are very different.
Yes. Yes. If proteins are a little bit similar, but if we're looking at sheets, they're all very different. Well, so yeah, one could define it a different way. So usually in the protein folding, people use a P and Q. People typically use the contact number. So it's like 1,000 dimensions. What can you do? So usually it's like how many contacts you made. So it doesn't really matter where they are. It just depends on how many of them form a bond, something like that. So basically reduce that to 1 or 2 dimensions to the ones you can see. It's more relevant. Yeah, usually sometimes it's a little bit like entropy in one dimension, and one is energy. So like it's basically the structure, like it's a little bit like the length, the size of a molecule to some extent, and how disordered they are. They're not necessarily well defined, but yes, it's a little bit abstract, right? But to reduce them to these, people can sort of, in people do simulations, they use cube. Well, we can use anything because we don't really have a number, but it just tells you if you pick reduce to one dimension. So pretty much, like in our case, it's end-to-end distance, right, for a molecule, right? I don't really know what they are, but the molecule, its conformation is defined by the end-to-end distance of molecule. So that's one. The other one here, it's we can use force. It's not necessarily the other one, the one I drew, but in principle, when you use force, that means it's another one we can measure, and that tells us something about the molecule. So it's whatever it is that reduces to force. And then when people do simulations and they have something, they reduce to something they can calculate. Right. And then, of course, so there's a lot of debate that some people don't like the one the other one takes. Yes.
Start out with the molecules aren't bound to your gold substrate. You just sort of sit them there, and then maybe just sitting on the surface. And yet, I look at these curves, and you have the sawtooth pattern, and there's obviously some strong force holding the molecule on the surface that you're pulling against, or else you lift the molecule off the surface rather than snapping it apart.
Okay, so usually, let me see if I have. Usually in the beginning, sometimes it's clean, sometimes it's not. I have in the beginning is here. Of course, I'm hiding them here. Here, there's a lot of things you have to lift them off the surface. There's some strong, so-called non-specific binding here. There's a lot of things, and then it's juggling, and off the surface. And these well-defined things is the domain unfolding, each domain. So this is the initial thing to lift the whole thing off the surface. And this unfolding depends on the structure of the molecule, the interaction within the domain. This is the molecule and surface interaction, so we're laying down there, right? So it's these are not reproducible, but these sawtooths are reproducible.
Okay, so I think the next one.
Maybe the two molecules stretching. How can we make sure that only one molecule is
Right. So we have to worry about this in the beginning. So the first thing is if we have low enough density of a molecule, then the chance you get two at a time is small. So we intentionally make the density low, so our success rate, so we randomly pick, it's actually less than 5%. Most of the time we pick up nothing. And then another thing is if we happen to have two, we'll see, because we see them stretching and one will fall off, we'll see a big peak force drop, and then another one starting over. So if we have two molecules, somewhat obvious. And first we want to reduce the chance that happen, and if it does, we'll know and we'll eliminate those. Okay.
So you're basically, what I'm a little bit flabbergasted by is intuitively I would think of having much smaller centers area on the chip, and therefore the non-specific binding is going to be less, that part of the molecule is attached to the surface. So how do you actually overcome that initial so that little bit of attachment?
Well, first it's probably going to be smaller, but then the molecules sitting on the surface would not really necessarily completely flat down. So they are probably sitting, like some of them may not touch the surface because they're like a globular, right, sitting on top. So we're pulling on those. But if they happen to be stuck tightly, we may pull just, say, if we have 8 domains, maybe we only pull 2 domains out, and then it breaks. So that happens all the time. Right, so we don't rely on 100% perfect situation. It's just if we see signal, that's enough. If we don't see it, we're out. Right, that's the ones that's successful. Well, first. Okay, I don't do optical tweezers, but I can tell you some principles. Optical tweezers usually they have two beads, and they functionalize the bead with there's a receptor and ligand, usually biotin or streptavidin, and then that's functionalized, and then the DNA, and then there is another functionalized. So usually they rely on DNA handle. So if you have a DNA, the B is functionalized with a receptor, and then there's a ligand, and then another piece of DNA is functionalized as a ligand. So the ligand is going to bind to the receptor, and then so the DNA now is attached to the B handle, so-called handle. Usually, I don't know, it's a few hundred to 1,000 base pairs. So now the B has a handle, and then now your DNA comes, so that's complementary to the handle, will bind to that. So the optical tweezers, usually the binding, it depends on the handle. Usually they're not as strong as the non-specific binding. They're specific, but usually they break on the maybe around 100 piconewton, they'll break. So you see optical tweezer signal, usually they stop at about 100 piconewton.